I'm going to be talking about a tool that we're developing called phRAIDER, standing for Pattern Hunter-Based Rapid Ab Initio Detection of Elementary Repeats, which is basically, in short, is for de novo repeat identification. This is joint work between some of my master's students, Carly Schaefer, Nathan Figueroa, Zhao Liu, and then I am faculty at Miami University. That, however, is a little bit misleading. When you come from Miami University, you always have to specify that you're from Miami University in Ohio. Okay, we're located near Cincinnati. Every year I review applications for faculty positions, and I will always toss a few because in the cover letter they say, looking forward to your great weather and beaches. Yeah, doesn't work. All right, this is real quick a methods talk. The interesting things in this talk to me as a computer scientist are the mathematical model and the algorithm, but I realize that I'm not talking to a computer scientist, and even if I were, I couldn't cover that in the time limit. So what I'm going to talk here is kind of sketch out the method we are using for de novo repeat identification and compare it to other tools to show you that we've got a tool that is working as well or better than anything else. We are, so we're looking for to identify genomic repeats. We just had a talk about L1s. We're not focusing on those specifically, but more just any repeated data. We're interested in these for a number of reasons. I work with them in an independent project, using them as a window to evolutionary history. We can, by looking at them reconstructing ancestors, we can determine things about the history of neutral substitution rates, that sort of thing. They're also a source of genetic disorders. They are known mechanisms of evolutions. There's evidence that they have, for example, created exons. And then the, to me, least interesting, but probably most important thing is that you want to be able to mask them out. If you have a genome that you want to apply some prediction or annotation tool to, you probably want to mask out the repeats first, and your tool will work much better. And as we've just seen, they are a major component of most eukaryotic genomes. They're, 45% of the human genome. I think corn is something like 90%. All right, so there's something that are really important to be able to identify. Now, if you've worked with this at all, you have probably run across the tool REPEATMASKER, which is the go-to tool for identifying it. The problem with REPEATMASKER, well, not the problem, but the thing you need to know about the REPEATMASKER is that it's a library-based tool. If we think of, let's take transposable elements as families. All right, a family is all members descended from a common ancestor, one common ancestral sequence. REPEATMASKER needs some sort of description of the family, probably an estimate of the ancestral sequence, and then it can find more members. So if you have that library, REPEATMASKER is the tool to use, but it cannot discover new families. And if you have, say, a newly sequenced genome where you haven't developed the library, and you don't have any closely related sequenced genomes where you already have a library that may be applicable, then you have to find, you have to have some way of finding the initial family, creating the library. All right. So this is the problem of de novo repeat finding. Given an unannotated large genomic sequence, find every subsequence of some minimum length or longer that occurs at some frequency across the genome. We're not, of course, the first people to address this. There are a number of tools out there taking different approaches. One obvious approach is just self-alignment, using something like Smith-Waterman to align the sequence to itself and look for common subsequences. The problem with that is that it's just not scalable to a whole genome approach. Other tools use string matching algorithms. They use data structures such as suffix trees or suffix arrays, right, to find common substrings. The problem with that is that they don't account well for sequence variation. They're not generalizable to the problems we have with the repeat sequences not being identical due to substitutions, mutations, whatever. Third approach I'll mention real quick is the de Bruin approach. If you are at all familiar with sequence assembly, okay, you've seen de Bruin graphs. This is a variation on de Bruin graphs Pavel Pesner came up with, and once again, though, it's not really scalable to huge inputs. So there have been a lot of tools in these different categories. Under self-alignment is Recon and Pyler and others. In the de Bruin approach has Repeat Gluer. And then under the string matching approaches, which is where we fall, is Reputer, REAS, and most significantly, Repeat Scout. There's sort of a consensus in the literature that Repeat Scout is the prime tool. If you're only going to use one tool, you're probably better off using multiple tools, but if you're only going to use one, Repeat Scout is the tool to go with. And so this is our basis for comparison in our tool. So to describe our sort of high level of our approach, our first shot at this was a tool called RAIDER. rapid ab initio detection of elementary repeats. Now notice this phrase, elementary repeats. This tool was not actually looking for full repeats. They were looking for what we conceive as sort of a building block of repeats. These were first proposed by ZHENG and LONARDI, and the definition went like this. Given a threshold L, and a length threshold L, and a frequency threshold F, an elementary repeat is a subsequence A such that, first of all, A is long enough. So we're going to disregard anything less than length L. Second, A has a sufficient frequency. It occurs at least F times across your genome. For us, F has been 2, but our software is designed to accept higher values. Here's the important one. A contains no subsequence B such that B is long enough to be considered elementary and occurs independently of A. So you have your multiple copies of A across the genome. Each of them contains a subsequence B. B cannot appear anywhere else. And finally, these have to be maximal. An elementary repeat cannot be contained within something else that also meets the definition of an elementary repeat. So they're sort of as large as possible. So this is sort of a mathematical definition. One of the drawbacks to it is it doesn't really account for sequence variation, which is where we're going with this. Okay. And there had been other algorithms, or there had been previous software to detect them. The problem is that most of the software was either quadratic runtime, so they weren't really scalable, or really couldn't deal with variation. All right. So we came up with RAIDER. One of my master's students, Nathan Figueroa, came up with a linear-ish time algorithm of elementary repeats. I say linear-ish because as a computer scientist, I can't say it's linear in the worst case, but in practice it really is. All right. The more important thing, though, it's amenable to a pattern hunter space seed strategy. If you're not familiar with this phrase, I will explain it in just a minute. But basically, it takes a parameter called a seed, and over here we had the initial results. The only thing to really get out of this is that, so we tried RAIDER with several seeds on human chromosome 22, and we see where Repeat Scout takes about 2,300 seconds, RAIDER takes at most 192 seconds. How good is it? Well, we managed to cover, if you look at the actual consensus sequence we were searching for, Repeat Scout covers about 77% of it, we cover 84%. Down here, we see other chromosomes, and while we are still much, much faster, the results over in the coverage aren't so good. And our conclusion was that RAIDER this approach had potential, much faster than Repeat Scout, and had the potential to beat out Repeat Scout. And furthermore, this idea of using these seeds works. As you change the seeds, you get different results. All right, so let me explain what these seeds are. Pattern Hunter was a proposal, was a tool, came out somewhere around 2002 by Mod, Trump, and Lee to improve BLAST. So the original purpose was to make BLAST much better. And it works by where BLAST, or it's appropriate in circumstances where a string requires, where you're looking for common substrings. If you remember in BLAST, if you're comparing, say, a query sequence, query sequence to some target sequence, before you align them, you look, do they have a common substring of length, usually 12? If they do not, you dismiss them. Otherwise, you go ahead and spend the time on the alignment. Well, with sort of a special case like this, where we have a string of length 23, which is identical in every base except this one, but this is exactly longly placed, so you don't have a substring of length 12. So what Pattern Hunter says, instead of looking for 12 consecutive matches, have some sort of structure to where you're looking for the matches. For example, maybe we look for 6 matches and 6 matches with one base in between where we don't care if it matches or not. All right. So you can line this is the seed, you can line it up, you get A to A, C to C. Everywhere there's a 1, you have matching bases. This would pass then the Pattern Hunter filter in BLAST and allow you to align them. This has proved to be incredibly effective. It greatly increases the sensitivity of BLAST without really adversely affecting the runtime. And the concept of seed, this is one sample seed, but there's all kinds of things you can use as a seed. Basically, there's some structures to where you want matches and where you don't care about them. Okay. We wanted to figure out how to use these. And so I brought my next graduate student came in, Carly Schaefer, and she started messing around with it. RAIDER had it, but it wasn't done very well. It was sort of a hack. She thought about this more, and what she realized is that you can include these into the definition of the elementary repeats, but they require some changes. So it required a generalization of the repeat element definition to incorporate seeds, which if anybody is more mathematically oriented is the part that really interests me and is in the appendix of our proceedings paper. And and it required a reworking of the algorithm to accommodate the new definition. All right? And thus we came up with phRAIDER, Pattern Hunter-Based RAIDER, a linear-ish algorithm for identifying all transposable elements with respect to a single seed. If anybody's worked with Pattern Hunter, there are two obvious questions here. Let me answer them now. First of all, Pattern Hunter you usually use with multiple seeds. We are not there yet because that requires the restructuring, is going to require the restructuring of the algorithm and the definition again. We're not quite sure how to deal with that. I've got a student working on it. There's also this question of what makes a seed good. When you use Pattern Hunter in BLAST, you want to be careful about what seed you pick, and figuring out what a good seed is not easy. It's actually a really computationally challenging question. Here, the model is somewhat different, and we don't fully understand it. So at this point, we have no idea, other than just trial and error, what makes a seed good. So let me go ahead and skip to the results. The algorithm for this is described in the paper and more thoroughly in the appendix. But in terms of the results, how did this turn out when we implemented this? So first of all, let's look at resource usage. What I have over here is a plot of runtime. I ran it on individual chromosomes, going up to 250 megabases. Okay. In red, we have the runtime for repeat scouts. In blue, we have the runtime for RAIDER, significantly better. The one problem we have with RAIDER is memory usage. It's a real memory hog, so we had to come up with a memory efficient version, which is the green. We sacrifice a little bit of time to get better memory usage, but obviously still much better than repeat scouts. Over here, we have that memory usage, and we can see the RAIDER is really hogging up the memory. We are unable to, say, run this. We had access to a machine with 180 gigabytes of memory. We were unable to run it on the human genome, but when we used the memory efficient version, we were fine. All right. The next question is, do these seeds matter? I'm claiming having these seeds will help with respect to sequence variation. So, and what is a good seed? We kind of messed around a little here. Over here, we took seeds that didn't have spaces. So here we were really looking for just consecutive matches. The question is how many? We let that range from 20 to 100 using two metrics. The red indicates how we did when we were only trying to mask out the individual bases, if that's all we cared about. The blue indicates how we did if we wanted to use our output as a library for REPEATMASKER. All right, so produce our output, run it through REPEATMASKER, see how well did we do in actually identifying repeats. And what we see for the red is that as the seed gets longer, we do much better. This dotted line here shows how Repeat Scout does. Repeat doesn't take a seed, so there's only one run. Okay, and clearly we're doing much better. However, for the actual transposable identification for REPEATMASKER, we don't do quite as well here until we actually introduce our spaced seed. So we have this concept of density. If we had a speed that was, say, required 32 matches, but had 64 characters, so 32 wildcard characters, we'd say the density was 0.5. And we notice when we start actually lowering the density, we start doing better. We're doing better with the masking metric either way, but here around a density of 0.7, we do better with the, our tool starts beating out REPEATMASKER. Sorry, Repeat Scout. Sorry, I lost my train of thought. All right, so this looks promising. We started running on actual chromosomes. We had a little bit of a problem. We couldn't run this, at least at the time, on very large chromosomes because while our tool is quite fast, the analysis tools, we had to use BLAST, we had to use REPEATMASKER, take quite a long time. We couldn't do hundreds of thousands of runs like we wanted to because of those. But we ran them on different chromosomes. And basically what to read from this is that in terms of the masking sensitivity, how well do we do masking things? We're pretty consistently beating out repeat scouts. In terms of using it for a REPEATMASKER library, we're not doing quite as well, but we're not that far off. That's just about. And we are getting huge speed ups over Repeat Scout. You know, here we get 260 times. Here we're over 2,000 times speed up. So what we've got is a much faster tool, which is getting quality results, depending on which metric you want, better than or almost as good as Repeat Scout. All right. So I think we're about out of time, but we are continuing working on this, and so what I've just said is what's discussed in the Proceedings paper. But we are now we've now just finished the next step, repeat element assembly. You get these repeat elements, and now you want to assemble them into actual transposable elements. All right, so repeat elements are supposed to be the building blocks of genomic reads. They need to be assembled, and so our tool, currently called phRAIDER CD, unless I think of a better name, is doing that. And what we're finding is that this assembly step takes a little bit of extra time, but really not much. And it is a significant improvement on result quality. When we then use that as our library for Repeat Masker, we get results which are at least as good, if not better, than Repeat Scout, with overall runtime still orders of magnitudes faster. That would be my time. All right. So this is an open source tool available off of GitHub. It's cited in the paper. We're still it's still in development. That phRAIDER CD, the manuscript is in preparation. We'll release that when we've got it. So I'd like to acknowledge on this. This is really the work of 4 people. Carly Schaefer was a master's. This was her master's. The second part of this was her master's work. She's now at Google, but will hopefully, a fantastic student, hopefully will be coming back for a PhD at some point. Nathan Figueroa, also a master's student, now GuideWire, and Jalin Liu, who was working on that assembly problem as part of his PhD, though who is currently interning at Google, hopefully will come back. All right, so thank you.